Unit 2.1 to 2.3, Cell Structure and Function. We will start by taking a tour of the cell. These are things you must know. This is considered in scope with AP Biology. Please review these so your studying can happen strategically. We'll start with two general categories of cells. The first category is prokaryotes. These are primitive cells from the domains bacteria and also archaea. The second category are eukaryotes. All other domains have cells that are eukaryotes, including protists, fungi, plants, and animals. Below is a picture of a prokaryote. And here is one example of a eukaryote, more specifically, cells from the lining of a human uterus. The stained purple picture in the animal cell is of the nucleus. Specifically, the genetic material within the nucleus is stained in this picture. Note that there is no nucleus in the prokaryote. This is a diagram explaining the anatomy of a prokaryotic cell. Please note the absence of a nucleus and instead what is called a nucleoid. Oid, that suffix means resembling. This resembles a nucleus. We can see the genetic material kind of bunched up here. However, it is not a nucleus. There's no nuclear membrane surrounding the genetic material. Other common features of bacteria include extracellular features such as fimbriae or cilia or flagella, and organelles such as ribosomes. There is a plasma membrane and even a cell wall. In this slide, as I click through it, will show a compare and contrast between prokaryotes and eukaryotes. Prokaryotes, again, have no nucleus and instead have their DNA in a nucleoid, resembling a nucleus, but there's no nuclear membrane. You saw ribosomes in a prokaryote. There's no other organelles, only ribosomes. They're small, they're primitive, and they're in simple organisms like bacteria and archaea. Compare that to a eukaryote, which has a nucleus and a nuclear envelope or membrane. There are many different organelles in a eukaryote, as we'll see very shortly. They're larger, more complex, and are considered more advanced than a prokaryote. Examples are plants and animal cells, which we will view in this slide deck. The nucleus. Its function is a control center of the cell. It contains the genetic material of the cell, specifically nearly all the DNA. We'll get into a couple examples of DNA out of the nucleus, but for intents and purposes, the genetic material is found in the nucleus. Messenger RNA is also transcribed in the nucleus, so you'll find mRNA in there too. This is important to mention because not all organelles contain a double membrane. Yes, there is a membrane surrounding the nucleus, but it is a double membrane, commonly referred to as the nuclear envelope. It's continuous with a structure called the rough ER or rough endoplasmic reticulum, which will be discussed later in the slide deck. But when I say continuous, what we mean is adjacent to. They're connected. They work together. The nucleus is porous, so materials can enter the nucleus and they can also leave the nucleus. Consider protein synthesis. you don't build proteins in the nucleus, so directions from the genetic material have to exit the nucleus. That's just one example of materials entering and leaving the nucleus. The genetic material, most of the time, is found in a state called chromatin. When I say complex, I mean combination. And it's DNA and proteins together, and we'll see pictures of that shortly. These get wound up to make chromosomes during cellular division. A region in the nucleus called the nucleolus is where ribosomal subunits are formed. We will get into ribosomes and their structure and functions very shortly, but just understand that there is some protein synthesis going on here. Specifically, ribosomal subunits are being made. Here's a picture of a nucleus from a electron microscope. And here, even more zoomed in, are the pores surrounding the envelope of the nucleus. Shown in a model, you can see the nucleus, the nucleolus region, the chromatin filling the nucleus, a membrane around it. This is a double membrane. If you look closely, you can see the double membrane. Pores on the surface of the nucleus and an adjacent structure called the rough endoplasmic reticulum. Closer, you can see pores on the membrane. Also ribosomes on the surface. And here is chromatin, that genetic material, that DNA this chromatin not wound yet, but loosely in the nucleus with these proteins that aid in wrapping the genetic material during cellular division. More on that in later units. Ribosomes. The function of ribosomes is to synthesize proteins. So where is a polypeptide assembled? It is assembled with a ribosome outside of the nucleus. Since all cells have proteins, or since I should say all organisms have proteins, all forms of life will have these protein synthesizers or ribosomes. That's why you find them in prokaryotes and in eukaryotes. Specific structure, this is a complex of proteins, so there's a large subunit and a small subunit. We can classify ribosomes into two different categories, one being free ribosomes, which will float in the cytosol or cytoplasm. And then there's these bound ribosomes that attach to the endoplasmic reticulum. They have slightly different functions in which proteins they produced. Intracellular proteins are constructed with these free ribosomes. They'll just come in contact with messenger RNA strands and read those strands to assemble polypeptides. Bound ribosomes in the endoplasmic reticulum will make proteins that will eventually leave the cell, and that transport is a huge part of this slide deck. So we'll learn the players involved with that transport. Picture of a ribosome, both microscopic image and a model showing the large subunit and the small subunit. This is that transport system I was talking about. Looking at the model, you see the nucleus, you see the adjacent structure, the rough and smooth endoplasmic reticulum. Let's see, did I cover? No, I didn't cover ER yet. That's great. And a new structure, which I've yet to even mention yet, is the Golgi apparatus. So you can see movement with these arrows, materials exiting the cell. This is the cellular surface right here, this plasma membrane. Here's the endoplasmic reticulum, otherwise called ER. It's a network of membranes and sacs. Just going back to the picture, we're right here with the endoplasmic reticulum adjacent to the nucleus. Two categories or two areas of the endoplasmic reticulum are the rough ER, which has ribosomes on its surface. Think of the purpose of ribosomes, aiding in protein synthesis. So the rough ER will package proteins for secretion. Remember, protein synthesis occurring on the rough ER is meant to be exported out of the cell. The Golgi helps with that. More on that later. Compare that to the smooth ER, which lacks ribosomes on its surface. Since it's lacking ribosomes, it will not make proteins. The smooth ER's function, rather, is to synthesize lipids, also metabolize carbs, which means to break down those carbohydrates. It can also detox drugs and poisons and even store the calcium ions. Huge purpose for calcium ions will be discussed later on in the class. Pictures of the endoplasmic reticulum. Notice the smooth ER lacks ribosomes, whereas the rough ER contains ribosomes. You can even start to see some materials leaving the rough ER in what is called a transport vesicle. Also a great shot of the nucleus and its pores leading right into the ER. Another player in this transport system is called the Golgi apparatus. Let's look at a picture of all of it together real quick. Here we're going to talk about the Golgi. So notice materials leaving the ER and entering into the Golgi apparatus. So the Golgi apparatus will receive polypeptide structures and ensure that this folding, so going from this primary structure to the tertiary structure and even beyond, this is all conducted in the Golgi apparatus. So they'll ensure correct folding and modifications of newly synthesized proteins. And they'll also get them ready to move out of the cell or around the cell. These are also flattened membrane sacs. The biological term for them are called cisternae. An important distinction between the two sides of the Golgi apparatus. The cis face is what will receive vesicles and receive polypeptides from the rough ER. And the trans face will be on the opposite end of the Golgi and will export out folded proteins. Again, the mechanism of shipping is these compartments called vesicles. And after reaching the Golgi, proteins are essentially ready for export. So it is at this point in the slide deck where we shift our focus a little bit from membrane transport and more toward maintenance. And we'll examine the lysosome. The function of a lysosome is maintenance, intracellular, within the cell, digestion, recycling a cell's materials, and even governing cell death, apoptosis. More on this when we cover cellular cycle. How does a lysosome perform these functions? Well, it utilizes the help of proteins, specifically hydrolytic enzymes. Think of the word hydrolysis, and then compare that to this word, hydrolytic. Pictures of lysosomes, two examples here. You can see phagocytosis occurring on the left model, where a food vacuole enters the cell and a lysosome aids in this digestion. You can also see a vesicle containing a mitochondrium and also a structure known as a peroxisome, which we will discuss its purpose later. But here in this autophagy, the lysosome is actually digesting and recycling old cellular materials. So these are great words to know, phagocytosis and autophagy. And just they apply, these are the purpose. This is what the lysosome does. More on the maintenance of cells, specifically with storage, are vacuoles. Vacuoles have a wide range of storage ability, including food and water, minerals, pigments, and even poisons that are not supposed to be elsewhere in the cell. These have a membrane around them, and there's that term vesicles again. So almost these containers. Examples include food vacuoles, contractile vacuoles, and a great example are in plants. These plants, this is a hallmark of a plant cell. They have a large central vacuole which stores water and some ions. This is not in an animal cell. It is unique to a plant cell. And maybe think about how that is observed in the structure and function of plants. Going back to the transport one more time, starting in the nucleus, genetic materials like mRNA can leave the nucleus. Materials are received by the endoplasmic reticulum, where proteins are synthesized by ribosomes. This is where your polypeptides get built. The cis Golgi receives polypeptides from the rough ER. And also don't forget the smooth ER, lacking ribosomes, but participating in lipid synthesis and other functions. Back to the rough ER, vesicles will leave the rough ER carrying polypeptides to the cis face of the Golgi. The Golgi will receive these polypeptides and will participate in folding them and monitoring that folding and modifying that folding. Proteins are folded in the Golgi and leave the transface to either be circulated within the cell or leave the cell. How is all this powered? This requires energy. Cells require energy to function. That energy is supplied by the mitochondria, which is often referred to as the powerhouse of the cell. But here, stored energy is released into chemical energy. That process of accessing stored energy and using it is called cellular respiration. And we will cover the mechanism of cellular respiration in great detail later on in the course. This is another organelle with a double membrane. That's very important to know. In fact, there's vocabulary showing specific areas of a mitochondrial membrane. Examine these folds. We call them cristae. And these folds are really important because they increase the surface area so we can maximize ATP production. And this is where this is your energy molecule, it's adenosine triphosphate, ATP. The fluid within the mitochondria is called the matrix. Chloroplasts. These are organelles found in plant cells, and their function is to conduct photosynthesis, which will be discussed again in great detail when we look at some cellular processes. And our understanding can be right now that photosynthesis harnesses the energy from the sun and ultimately produces glucose. Again, double membrane in chloroplasts. Some terminology with chloroplasts. You see these discs inside the membrane of the chloroplast. These discs are called thylakoid discs. When you stack these discs together, and they are often found, pretty much exclusively found, stacked together, and we call these stacks grana. The fluid inside the chloroplast, around the thylakoid discs, around the grana, is called the stroma. Within the thylakoid discs are pigments such as chlorophylls. That's the most popular that's the most popular pigment, producing that green color, capturing a particular wavelength of sunlight's energy. This is a fascinating theory that is used as evidence of common ancestry and provides a link between prokaryotes and eukaryotes. It is believed that mitochondria and chloroplasts were actually independently functioning organisms that were engulfed by a eukaryote. So if we follow these arrows, we start with the ancestor of a eukaryote cell, and over time, mitochondria were engulfed, and also over time, chloroplasts were engulfed, leading to a cell with much greater specialization and function, capable of more than its ancestor. What supports this theory? So this is the theory, and here's the evidence. Both the chloroplast and the mitochondrion contain a double membrane. Furthermore, mitochondria and chloroplasts have their own ribosomes and even their own DNA. That's right, you inherit DNA in mitochondria from your mother. Mutations in that DNA can lead to many genetic disorders, and you may discover that in your genetic disorder poster research. Even furthermore, in order to reproduce a mitochondria or a chloroplast, it is not necessary to reproduce the entire cell. So you have these organelles that have a double membrane, much like a normal cell has a double membrane. It has its own ribosomes and even its own genetic material, and it's capable of reproducing independently from normal cellular division. Returning to maintenance with our final organelle of the slide deck are peroxisomes. The function of peroxisomes is very similar to the overall function of a lysosome, but it contains a different material. So instead of the hydrolytic enzymes, you're going to find hydrogen peroxide inside a peroxisome. And hydrogen peroxide assists in breaking down fatty acids and also detoxifying alcohol. Overall picture of a eukaryote. Nucleus with the nucleolus region inside it, porous surface adjacent to the rough ER and the smooth ER. ribosomes coding the rough ER, receiving genetic material exported by the nucleus, synthesizing polypeptides, the Golgi nearby receiving polypeptides on its cis face, exporting on its trans face, and in between folding the polypeptide, essentially building proteins. Maintained by the peroxisome with its hydrogen peroxide and the lysosome with its hydrolytic enzymes, powered by the mitochondria holding cellular respiration, producing ATP, notice similar structures in a plant cell: nucleus, endoplasmic reticulum, smooth ER, Golgi, mitochondria, peroxisome, peroxisome, and unique to the plant cell, cell wall. You do not see this in an animal cell. Chloroplasts with the thylakoid disc, the grana, the stroma, and a large central vacuole. Please note also that there are no lysosomes in plants. There are no centrioles in plants, and we'll talk about centrioles later. And some plant sperm have flagella. Again, we'll get into flagella later. You see this word elsewhere in the lecture, plasmodesmata. That's just a porous surface on the cell wall. That's where that word comes from. This is important. Make sure you recognize that there are differences between plant cells and animal cells. And it goes beyond just having a cell wall and chloroplast. There's some other critical differences too. We're going to shift and now discuss cell size and scale. So we'll consider why we can't have giant organisms with cells the size of footballs. Why can't that happen? There's a great answer. There's a true reason why we don't see that in life forms, ever, and will never. First, just a picture showing scale. So our unaided eye can see just about to a human egg cell. Not quite, though. And with microscopy, you can see smaller items. And our cells are right in this range. Well below a millimeter. You can't view it with an unaided eye. Also notice here, my goodness, the length of some nerve and muscle cells are actually very, very, very long. But this is where we want to consider right now. Here's your answer. Why are cells so small? Well, think of the function of cells. Export and import. It's important to have a large surface area to volume ratio. When you have a large surface area, the rate of chemical exchange can be much more rapid than without it. Also consider the movement of materials within a cell and how far they would have to travel if the volume ratio was much greater than the surface area. This is a graphic explaining surface area to volume ratio as we trend toward a greater ratio. This is just a title slide or a slide containing effects that membranes and membrane-bound organelles, localized metabolic processes, and enzymatic reactions. This is a pretty dense statement explaining what we already talked about. Consider what metabolic processes are and enzymatic reactions. These are actions that happen in cells. If someone were to ask you what's the job of a cell in its most general terms, well, it's to it's to metabolize things, and it's to perform chemical reactions with the help of enzymes. So these membranes allow the space and the structure for these jobs to occur. For example, having membranes within around your organelles really allow for reactions to happen in a very localized area instead of being exposed to other competing reactions occurring elsewhere in the cell. Finally, internal membranes also increase surface area. Consider the mitochondria. I brought up surface area with mitochondria and how that really maximizes ATP production. Here you see surface area increased with the endoplasmic reticulum and also the Golgi apparatus, all these folds. And just another example of surface area, a really popular one, helping in digestion. Here we're looking at an intestine, specifically the small intestine, and look at all these layers. Think of the role of the small intestine in that absorption of materials. You'll want to maximize that. So that's why this membrane, this inner membrane, really has a lot of surface area. Here's what I just said in the small intestine. We can go from villi to even microvilli on the surface of this inner membrane. Another example are plant roots. Again, improving and really raising that surface area to volume ratio. Helps maximize the rate at which cells can do their jobs. These slides are review, just localizing information about cellular components, their structure, and their function. View these on your own. Great summary slides for what is discussed in this slide deck. And this moves on to old slides, which are accessible to you, but will not be discussed in this audio recording.